This mini lecture is going to be all about how organisms are classified. And you may have learned about this taxonomic hierarchy in other classes. This is a way of organizing organisms that are more similar in groups. If we start from the very bottom, species would be the ones who are the exact same kind of organism. And then similar species are grouped in the same genus, similar genre are grouped in the same family, similar families are grouped in the same order, and so on and so forth, all the way up until domain. And if you remember, the three major domains of life are eukarya, archaea, and bacteria. Kingdom is reserved only for eukaryotes, and so prokaryotes don't have kingdoms. They jump straight from domain to phylum. Let me show you an example using E. coli. Here you can see that the domain for E. coli is bacteria, right? That's the very largest group. All bacteria belong to the domain bacteria. There's no kingdom. E. coli are in the phylum Proteobacteria, and as you work your way down, you see that Escherichia, the E in E. coli, is the genus, and coli is the actual species. And then what about strains? You see over here where it says plus or minus strains. So E. coli, for example, can have what are sort of like subspecies where they are the same species. They're genetically very, very similar, but there are some small differences. Maybe they have a few different genes here and there, and this is what makes some E. coli strains pathogenic and some E. coli strains non-pathogenic. The ones who are pathogenic have picked up a few extra genes that allows them to make toxins or otherwise damage the host cells, whereas the ones that are non-pathogenic don't have those genes. But other than that, they are still the same species. And so we refer to these small differences between different members of the same species, strains. Let's go back to the domain now and see, well, how did three domains come to be anyway? The three domains are shown here on what is called a phylogenetic tree. A phylogenetic tree is sort of like a family tree or an evolutionary tree that arranges different organisms based on how related they are to each other evolutionarily, or how similar they are to each other. And so you can see over here, bacteria are on their own branch off here. They are much more closely related to other bacteria than they are to archaea, which are here in the middle. And those, there's yet another branch here for eukarya. And these three domains were really determined by using ribosomal RNA genes, which are found in all of cellular life. And so the similarities and differences between these genes were used to show that archaea really truly are on their own branch, even though on the surface they look very similar to bacteria. Right, so the phylogenetic tree of life is based on similarity between nucleic acid sequence that encodes ribosomal RNA. What is ribosomal RNA? There are three major types of RNA: mRNA, tRNA, and rRNA. mRNA is the one you're probably the most familiar with. This is the messenger RNA that is copied from DNA during transcription and then read by ribosomes in translation. Here are three types of RNA. Messenger RNA is the one that you're probably the most familiar with. It's made from DNA during transcription, right? So here's your DNA, and in transcription, this gets copied into RNA, and this RNA is messenger RNA that will be read by ribosomes and used to make proteins. tRNAs also play a role in translation. And what they do is they are the ones that deliver the amino acid to the ribosome during translation. Right, and then the one that we're talking about in this lesson is ribosomal RNA. So ribosomes, remember, they have a large subunit and a small subunit. And both of these subunits contain ribosomal RNA as part of their structures. So ribosomes are made up of both protein and ribosomal RNA. Because all cells have ribosomes, they all have ribosomal RNA, and that ribosomal RNA is encoded for by ribosomal RNA genes. It's those genes that can be sequenced to see how similar or different the ribosomal RNA is in different types of organisms. And the more similar they are, the closer they get to put together on that phylogeny, and the more different they are, the farther apart they get put on that phylogeny. And so, but why ribosomal RNA? It's because all organisms have ribosomes and ribosomal RNA, and there's enough variation in that ribosomal RNA to make it useful to distinguish between species. So these two characteristics are what make ribosomal RNA so useful.